In this video, I'm going to give you a general overview of the immune system. We have both the innate and adaptive arms. So let's start with the innate, which is not specific. When a pathogen decides it wants to infect us, first it has to get through the first line of defense. And when I say pathogen, it could be a bacteria, it could be a virus, it could be a fungus, protist, worm, whatever tries to infect us has to get through our physical barriers first. So that's the first line: our skin, our gastrointestinal tract, our genitourinary tract, our respiratory tract. These are all mucous membranes and have defense mechanisms built in. You know, if, for example, something in our respiratory tract, we have cilia in our pseudostratified columnar epithelium that just pushes that mucus and pathogens up and out. And then if it goes down our gastrointestinal tract, you know, we'll fry most pathogens in our gastric juice of our stomach. So that's the first line of defense. If it happens to get through that line of defense, then we're still in the innate immune system, which means whatever pathogen it is, they're kind of going after all of them, is the second line of defense. And so once it enters the body, we enter the second line of defense where we have some immune cells that try to help us out. So we have some immune cells like neutrophils, macrophages, dendritic cells. These are all phagocytic cells. They typically have something called pathogen recognition receptors on their surface. An example, the main pathogen recognition receptor is toll-like receptors. And these will recognize things that are foreign on the pathogen that enters our body. And these foreign molecules are called pathogen-associated molecular patterns. So these are some patterns that are on the surface of the pathogens that aren't, on the surface of our cells. So our immune cells will recognize these through their toll-like receptors, and they'll go and they will phagocytize that pathogen and release cytokines like interleukins, tumor necrosis factor alpha, colony stimulating factor. So interleukins and tumor necrosis factor, they may go up to the hypothalamus and get our fever going. So, what makes us feel bad when we're infected is our immune system's response for the most part. So that fever, malaise, chills, body aches, all of that is secondary to our immune response. So cytokines are just these chemical messengers that a lot of white blood cells use. So like the interleukin-1 and tumor necrosis factor can go up to the hypothalamus and stimulate a fever. Colony stimulating factors may go into the red bone marrow and stimulate proliferation of more white blood cells, because if we have an infection, we need more soldiers to fight in the war. So anytime somebody has an infection, you do a white blood cell count with blood, and you see that the white blood cell count is elevated. And then you can even look at a differential and kind of try to figure out what kind of pathogen it is. For example, neutrophils really like bacterial infections because bacteria are small and neutrophils can eat small things like bacteria. And so those will specifically rise up with a bacterial infection. So if you do a white blood cell differential count, you can see that those neutrophils will be elevated. So just to recap that, whenever a pathogen gets in our body, it has this little signature on its surface called pathogen-associated molecular patterns. They're recognized by toll-like receptors from our phagocytic cells and some other cells in the body, and they will respond to whatever kind of pathogen it is. And then we have another cell that likes to participate in the innate immune system. This is kind of unique because it's a lymphocyte. So, we have 5 different types of white blood cells. We have neutrophils, eosinophils, basophils, monocytes, which become macrophages and dendritic cells, and lymphocytes. Lymphocytes, for the most part, play a role in the adaptive specific immunity. But in this case, some lymphocytes called natural killer cells, they basically just look for anything that doesn't have a self protein on its surface. So all of our cells in our body that are nucleated, basically every cell but red blood cells, have this molecular pattern on their surface. And it's called the major histocompatibility complex 1. So that tells our immune cells, leave this cell alone, it's one of us. And if a cell comes into the body, some kind of pathogen, and it doesn't have this MHC1 complex on the surface, then these natural killer cells will go and they will directly kill that pathogen. So they'll bind to the pathogen and they'll release perforins, which cause perforations in the cell membrane and cause it to lyse, or they could release granzymes, which cause the cell to actually commit programmed cell death. So that's two ways that the natural killer cells, and that they like to attack viruses and they like to attack tumor cells. We're always having tumor cells trying to take hold in our body. If we have a robust immune system, it never takes hold. So these natural killer cells play a big role in keeping cancer from taking hold in our body. So another aspect of the innate immunity is interferons. So let's say that a cell gets infected by a virus, then that cell will produce, it's called STING. They're just genes that will be expressed and released that have a paracrine function. So para means around, so paracrine function is it'll affect all the cells in that vicinity, and it'll alert those cells that we have a virus here and they need to upregulate their defense mechanisms, and this will help fight the virus. And it's interesting because fever actually helps our phagocytic cells become more active. It also helps interferons to be more effective. It also sequesters iron. So fungi and bacteria, they have to have iron to proliferate and make new cells. So if our body hides all the iron in our liver and spleen, then they don't have that particular raw material. So that's a big part of what the immune system does. Now, I put this kind of in between the innate immune system and the adaptive immune system because complement can kind of work both ways. It kind of integrates with both. So what the complement proteins are, they're these zymogens. They're inactive. They're flowing through our blood, so they're made by the liver. And it reminds me a lot of the blood coagulation cascade, where you have one thing that gets activated and it sets off this cascade of activating other proteins. So this has a series of non-proteins, and when it recognizes something, it will fixate. These proteins will fixate on to that pathogen, and then they'll activate other proteins until they get a membrane attack complex, which makes a big pore in the cell, and that causes the cell to die. So that's how complement works. And it also causes chemotaxis, which means that it releases molecules that attract other white blood cells to the area. So I'll come back to complement when we go back to the adaptive immune system, because they really like to recognize antibodies, IgG antibodies in particular. So that was the innate immune system. That's the first two lines. So our first line is our physical barriers. Second line are these cells that just anything that's a pathogen, it's not specific, it does its best against them. But if this doesn't work, we have to bring in our big guns, and that's our adaptive immune system. Our adaptive immune system is really powerful, but it usually has to kind of get sensitized first. So that means it has to see that particular pathogen first, and then it takes a couple of weeks to build up some antibodies. So it may not play a huge role in that first infection, but if you ever see that infection again, then these antibodies are ready to go, and you will never see a symptom, most likely. So let's talk about the adaptive immune system. It's specific. That means that its receptors in this immune system are specific to a pathogen. Our lymphocytes, the cells, the B lymphocytes and the T lymphocytes involved in the adaptive immune system, there's some gene recombination and shuffling that goes on that allows us to have billions of different receptors. So it doesn't matter what kind of pathogen we could see, we're always going to have a receptor for it. It's really cool because we only have about 20 or 25,000 genes. So we'd never have enough genes to make a billion different proteins. So that shuffling happens, after the cell, while the cell is developing and maturing. So it's really cool how that works. So let's start with the adaptive immune system, and we have to introduce a cell called the antigen presenting cell, APCs. So antigen presenting cells, that's your macrophages, that's dendritic cells that are a lot like macrophages, and that's your B lymphocytes. So these cells like to kind of get in areas that pathogens could enter. So sometimes we'll have some dendritic cells that are kind of fixated in the epidermis of the skin and places like that, and close in the intestines, and wherever bacteria may enter, we'll have a lot of these cells. And so a lot of times they're the first to come into contact with the pathogen. So when they phagocytize a pathogen, they bring it in, digest it, the lysosomes, the organelles, come and merge with them and form a phagolysosome. And they'll digest it, and then they'll put the antigen that our immune system can recognize on its surface on the MHC2 complex. So I'll put it on the major histocompatibility complex 2. So let's say we have a dendritic cell. It's just seen a pathogen. It gobbles it up, and it puts its antigen on its surface. Now, what it'll do, we have lymph nodes distributed throughout our body. Whatever the closest lymph node is, it'll take that and take it into the lymph node. Think of the lymph node as like a police station where all these lymphocytes hang out. Both B and T lymphocytes like to hang out in there. And so they get in there because they know those antigen presenting cells will bring in stuff. And so this dendritic cell is bringing it in, this pathogen into the lymph node, and different lymphocytes will kind of bump into it, and a lot of them will bounce off because they don't have that specific receptor. But at some point, you're going to have a helper T cell. So we have two main types of T lymphocytes, helper and cytotoxic. So these helper T cells will actually bump into it, and it actually sticks because it's the same exact T cell receptor for that antigen. So when it binds to it, it's going to activate that helper T cell. Now, I have to introduce another protein on the surface of the T helper cells. And this one isn't very intuitive. It's called CD4, and that stands for cluster of differentiation 4. And so this is a co-receptor. So here we have the T cell receptor that's specific to this antigen. And that antigen is in the MHC2 complex. So those will actually kind of come together, and then this co-receptor, this CD4, will then come on and really activate that T helper cell. So the way I remember this is if you superimpose a 4 into an H, it kind of works out pretty well. And so that just helps me remember that T helper cells are always CD4 cells. They always carry that surface protein, CD4, on their surface that's used as a co-receptor in this situation. So now, we have this T helper cell that's activated. While that's going on, neutrophils, if we have like a bacterial infection, neutrophils are going to phagocytize that bacteria. And then after they phagocytize the bacteria, they will release the free antigens. They often die, and they'll release the free antigens into the blood. And the blood and interstitial fluid and lymph fluid, that's all our humors. So this is part of our humoral immunity that goes this direction. So when a B lymphocyte, it's interesting, B lymphocytes, their B cell receptors are actually their antibody. So they'll have their B cell receptor / antibody on their surface. And these are specific as well. So when that specific antigen binds to that specific antibody, it'll activate the B cell. But the B cell it's like its first step of activation. Also, it needs 2 steps typically. So that second step is that T helper cell that's specific to that same exact antigen will come and interact with the B cell. And this is how it happens. So the B cell sees this free antigen, oftentimes released by a neutrophil. It'll bind to it. It'll bring it in through receptor-mediated endocytosis. So it's specific receptor bound, and then endocytosis. So, those pseudopods will wrap around it and bring it into the cell, digest it with the lysosomes, and then it'll put its antigen in the MH2 complex. And that antigen can be seen by this T cell receptor that's already been activated over here. And so it'll bind, and then when they bind and interact, that's when this B cell really kicks in. So these little red dots, these represent cytokines. And I'm not going to go into all the specific cytokines here, but cytokines like interleukins will be released, and they will go and they'll bind to receptors on the B lymphocyte, and it'll activate the B cell to proliferate. So when the B cell proliferates, it always kind of divides into a plasma cell and a memory cell. So a memory cell, this is good for if we ever see this pathogen again, we're going to be able to fight it before we ever feel a symptom. It'll take care of it. Those antibodies skyrocket really quick as soon as that pathogen enters the body. The plasma cell is going to get activated now. So the plasma cell is going to make antibodies. And that's what plasma cells do. B cells become plasma cells. Plasma cells are the only cells that release antibodies. Antibodies have nothing to do with the T cell side other than just the activation up here. So plasma cells, it's interesting, if you look at a plasma cell, it has extensive rough endoplasmic reticulum. If you remember from cell biology, endoplasmic reticulum is where the rough endoplasmic reticulum in particular is where our proteins are processed and made. So if you look at these cells, they're just chock full of rough ER because they're making so many proteins, so many antibodies. And so these antibodies will be released, and when they'll go and they'll find the pathogen and they'll coat the pathogen. It's called opsonization. And when this happens, these complement proteins recognize that and they get activated, and so they'll latch on and cause the antibodies themselves don't kill the pathogen. They just kind of make them look tasty for the complement proteins. So they'll tag them, and those complement proteins will come in and cause that membrane attack complex, or just kind of those antibodies may allow phagocytes to recognize them better and come and phagocytize them. Sometimes antibodies will just go and neutralize toxins and stuff like that too. So there's different things antibodies can do. But antibodies come from the humoral side. So remember, the humors are the fluids. So these are our blood, our lymph, our interstitial fluid. This is kind of where all this takes place. Now, let's go over here and talk about the other type of T cell called the cytotoxic T cell. On this side, whenever a cytotoxic T cell, we have T lymphocytes flowing through our blood. So the most abundant white blood cell in our blood is neutrophils. It's around 65%. And then second is our lymphocytes. So we have our T and B lymphocytes flowing through our blood, and they're about 25% of the white blood cells found in our blood. And about 75 to 80% of those are T lymphocytes. And they're called T lymphocytes because they mature in the thymus. Like all of our blood cells, whether they're red blood cells or white blood cells, they're made in the red bone marrow. But those immature cells, T cells, will go to the thymus and mature there. So that's why they're called T cells. And then fortunately, B cells, they mature in the bone marrow, so it kind of gives you a hint on where their maturation takes place as well. So just a little FYI there. But so we have these cytotoxic T cells, and they're flowing around, and say they come across an infected cell. And what they look for is every cell in our body, like I mentioned, have major histocompatibility complex 1. So let's say that we have a cell, maybe it's a infected cell in our throat. You know, maybe the Streptococcus pyogenes, strep throat has latched on there, infected it. Well, or any kind of viral infection. You can, most of the sore throats we get are viral. So let's say this virus has infected this cell in the throat, and then it takes it in, phagocytizes it, puts it on its MHC1 complex. And then, it doesn't really phagocytize it. That virus actually just penetrates the cell. And then once it's in there, its antigens can be put on the surface in the MHC1. And so our T cell receptors for our cytotoxic T cells, and think about that word, cytotoxic, it's going to be toxic to cells. This directly kills cells. You see, T helper cells are more of like tattletales. They just try to send these cytokines out. But cytotoxic T cells, they go and they do the job themselves. They attack the actual infected cells. Because think about it, if we have a viral cell that is a virus-making factory, we want to shut it down. So we've got to get rid of that. And if we have a tumor cell, we want to get rid of that before it multiplies. So these cytotoxic T cells are really good for those situations. So if you see this, it sees this antigen in the MHC1 complex that's just any of our tissue cells, it'll recognize it. If it's specific for it, it'll actually bind to it, and it will release the same things that its lymphocyte cousin over here, NK cells, release, which is perforins, cause perforations in the cell membrane, granzymes to cause the cell to commit cell suicide. So that happens there. So we have the T cell receptor that's specific to this antigen, bind to it on its MHC1 complex. And cytotoxic T cells always have CD8 proteins on their surface to act as co-receptors. So the CD8 is used here. So we have CD4 that's with T helper cells, and CD8 that's always with cytotoxic T cells. So when that actually happens, the infected cell will die either from the porphyrins or the granzymes. And whenever this T helper cell has also been activated, it releases cytokines that cause this one to proliferate. It's called clonal expansion. It makes the same little clones of that because we have that particular pathogen we're wanting to fight. So it'll make effector T cells that are cytotoxic and we'll have more of them, and they can go to that area and kill all the virally infected cells or tumor cells. And then we'll have memory ones that are ready for the next time we see this. It'll be game on at that point. So it seems a little redundant to have T helper play this role to activate proliferation of cytotoxic T cells or to activate the B cells, but this kind of double handshake situation really helps to prevent autoimmune diseases. So I know that was a lot. I hope that helped you understand the immune system, and I'll see you in the next one.